Today it's my great pleasure to talk with Dimitri Kullmann about epilepsy. He's an expert on the regulation of neural network excitability, what's happening at synapses, what goes wrong in epilepsy, not only in epilepsy, but he's done work on other neurological disorders, particularly ataxias. He might want to talk a little bit about that. He's a professor of neurology and clinical experimental epilepsy at the University College London Queen Square Institute of Neurology, and he's on the faculty of brain sciences at UCL. So welcome, Dimitri.
Well, thank you, Mark. I'm delighted to have this opportunity to have a chat. And thank you for inviting me.
Yeah, and so I guess before we start talking about epilepsy, talk a little bit about your background. So I gather you spent quite a bit of time your training at Oxford, right? Both undergraduate and PhD were at Oxford?
Yeah, so I started medicine at Oxford, and then the British system is mainly to do the preclinical part of medicine, and we start very early, so we don't do a pre-medical degree. So I started medicine at the age of 17, and then normally you'd go on and do the clinical part where you're actually seeing patients. But I broke that and did a PhD or a DPhil in the middle, and hesitated about going back to medicine. But eventually I did go back to medicine. And then I actually vacillated, went back and forth between basic science and medicine. So I did my DPhil with Julian Jack on spinal cord motor circuits. And then after I sort of finished medicine and did my, what they call the house jobs, the house position, I went back to Oxford and spent another year and a half doing some more work, writing up papers, doing some more experiments. And then I went back to medicine, and then I went to California. So I went to San Francisco and did a postdoc with Roger Nickel for a couple of years.
Yeah, so you start out in the spinal cord, and where maybe in some sense, maybe the circuitry is a little bit easier. Well, it is, I guess, a little less complicated than the brain. And then with Roger, you started looking at kind of the classical, what's thought to be the cellular correlate of learning and memory, long-term potentiation. Is that right? And looking at different aspects of that?
I sort of landed in the middle of the so-called LTP wars. So back then, that was the most important question. It was effectively the only question that people were interested in. Yeah. Is LTP expressed pre or post-synaptically? So there were some pretty big personalities working in this field.
Oh, yeah. And that's...
I put my oar in.
And I think that's about the time that the receptors for the different glutamate receptor genes had just been cloned, and so then it became possible to manipulate genetically.
Yes, I mean the genetic manipulation came later, but at that stage the pharmacology had just been worked out, and right. And yes, you're right. The AMPA receptors were cloned by, the glutamate receptors being cloned simultaneously by Peter Seberg and by Steve Heinemann. So there was a lot of progress on the genetics, on the molecular biology, but the pharmacology was being sorted out as well. And of course, Roger contributed an enormous amount to the pharmacology.
Yeah. Okay. So then did you go straight to UCL from there?
Yeah, so I came back to Britain on a Medical Research Council fellowship, which allowed me to set up my own lab. And I also completed my neurology training. So I did my residency in neurology, sort of in parallel with setting up my own laboratory. So finally, after vacillating back and forth, switching between basic science and clinical medicine, I finally found a way of doing both in parallel. And I still do a small amount of neurology. So it adds up to about a day or a day and a half a week. The rest of the time I'm in the lab.
Okay, and then epilepsy, I mean, most everybody's heard of epilepsy and probably know a friend or family member that has had seizures. And what are kind of the demographics of epilepsy? How common is it?
Well, epilepsy is really a collection of very large number of different diseases. And one of the things that puts people off is just the complexity of how to classify epilepsy. But overall, it affects about 1 person in 1,000, in 100. So about 1% of the population has epilepsy. And the incidence is sort of has a sort of U-shaped age distribution. So it's common in early in life and later in life. So early in life, it's a lot of sort of birth injuries, genetics, infections, and late in life, there's a lot of neurodegenerative conditions, vascular conditions, sort of dementia, and so, but it can affect any age, and it can start at any age. But as I say, it's about 1% of the population has epilepsy. And interestingly enough, the prevalence is thought to be pretty similar in different populations. And low-income countries also have about 1% of people with epilepsy, but the incidence is probably higher in some of those countries. And that's probably reflecting the increased mortality. So epilepsy overall does have an increased mortality compared to general population. And it's not only attributable to seizures, so accidents during seizures, and something else called SUDEP, or sudden unexpected death in epilepsy, is another entity that's increasingly recognized. But people with epilepsy have an increased, have lots of comorbidities, general medical comorbidities, as well as psychiatric.
Yeah.
So it's an important collection of diseases. But as I say, it's a collection of diseases.
So you mentioned that more common in children and then elderly, and I know that in patients with Alzheimer's disease, they have a big increase in like 20, 30-fold increased incidence of seizures. And so I guess it's a good time then to talk. Now, so there's a lot of famous people. I was looking this up a little. Actually, I've written a book, Dimitri. It's called Sculptor and Destroyer: Tales of Glutamate, the brain's most important neurotransmitter. And so there's a lot of famous people like Prince, the singer and performer. He had childhood epilepsy. And then there's a really good American runner, Olympic gold medalist, Flo Griffiths-Joyner. She apparently actually died from a seizure.
Right.
And so, and then also one of our presidents, of course, here it's just kind of not necessarily scientific, but apparently Franklin Delano Roosevelt also, when he got older, had some seizures.
Really? Yes. I mean, I suspect that a lot more famous people have epilepsy than we know, because it still does carry some stigma.
Yeah.
So, yeah, I think a lot of people don't want it to be known that they have epilepsy.
Oh, the stigma. So that historically, you mentioned Africa, right? And my understanding is that still some of the tribes there think that a person with epilepsy is like possessed by demons. And I know that was kind of you go way back thousands of years before Hippocrates, I guess, that people would have a seizure and they think they're.
Yes, although I like to think that in most societies that analogy is no longer used. But actually, some of the analogies that people use are still pretty sort of imprecise, and people talk about seizures being like short circuits or avalanches or earthquakes or volcano eruptions. And in fact, none of those analogies are particularly helpful. I guess the sort of short circuit, the electrical analogy, or the lightning bolt, at least it brings in some electricity, but in reality, none of these are particularly helpful analogies. Yeah. And in fact, I think Hippocrates, you mentioned Hippocrates, I think he actually said, because of course there's this concept of the sacred disease. Right. I think Hippocrates said there's nothing sacred about epilepsy. I can't remember exactly how he phrased it in ancient Greek, but he said that it's actually a disease like any other disease with a cause and manifestations. So he was way ahead of his time.
Yeah. Yeah. Here we are, I don't know, 4,000 years later?
Well, 2,500, isn't it?
Oh, 2,500. Yeah, that's right. Okay. All right. So what's the current understanding? And no, it's still not, I guess, are there particular brain regions that are affected by a seizure? or is it kind of random?
Well, I mean, that's an important distinction between different types of epilepsy. So one of the ways that one can sort of slice up epilepsy is into primary generalized and partial onset or focal. So focal epilepsy is where the seizures arise from a specific part of the brain, most typically in the cortex. So places like temporal lobe, frontal lobe, depending on which particular part of the brain is the site of the epileptogenic focus. And primary generalized, where the seizures arise simultaneously in both hemispheres. And that doesn't map perfectly onto the types of underlying causes, but penetrating brain injuries and tumors and strokes often cause focal epilepsy, and there's an obvious place at which the seizures arise. But primary generalized epilepsy is usually thought of as a genetic condition, although it's usually polygenic. That's the current model. So monogenic epilepsy is actually pretty uncommon, and most of the monogenic epilepsies are generalized, but that's not a hard and fast rule. There are some genetic epilepsies which cause focal onset seizures. So as I say, it is complicated.
Yeah.
But that was one of the things that sort of that's one of the revolutions in the field, is to identify those monogenic epilepsies, and that started about 20 years ago with a number of families that were identified where the gene was identified that was responsible for the epilepsy, and a lot of those were ion channel genes. And of course, as a synaptic physiologist, that really got me very interested. Although at that time I was already working on episodic ataxia type 1 and a few other rare movement disorders caused by ion channel mutations. So episodic ataxia type 1, or EA1, is a very, very rare channelopathy caused by mutations of a potassium channel, Kv1.1, the so-called Shaker potassium channel. And so that sort of link between ion channel physiology and neurological disease has obviously grown, and I've benefited from the explosion of new mutations, new genes in this area, and to some extent have contributed. Well, I must admit, I'm not a geneticist, but I have been working closely with some colleagues, especially colleagues here at the Institute of Neurology. So Mike Hanna, Henry Holden, a number of other people who've identified families, and then my lab has contributed by trying to understand what these mutations do. Are they causing a gain or a loss of function? And sometimes it's not so obvious. Mutations may have a deleterious effect on the ion channel itself, but it has unpredictable effects on synaptic transmission or neuronal excitability, because you always have to take into account other factors such as where does the channel normally go, which subunits does it co-assemble with, and so on, and which neurons is the channel present in normally, and so on.
You mentioned the Shaker potassium channel. That was actually identified when I was doing my PhD at University of Iowa. Changfang Wu, working in Drosophila, identified the homologue, and there's of course a human homologue, and of course loss of function of potassium channel. Potassium repolarizes the membrane, right? Potassium's high inside, then when the cell fires, potassium goes out, that quiets the neurons down. Yeah, so those flies essentially had seizures.
Yeah, yes. Well, I think the term shaker came from the abnormal movements that the flies have under certain conditions. So yes, in fact, Lily and you and the Jan lab, the Jans were working in San Francisco when I was there. So I got to talk to them occasionally. So we've done a lot of work on Kv1.1. So these mutations associated with episodic ataxia type 1 tend to be dominant negative. So the channel, the subunit co-assembles with other Kv1 subunits. So you typically have a heteromeric channel. So you have 4 Kv1 subunits and additional Kv beta subunits. So it's quite complicated sort of predicting exactly what's going to happen if an individual carries one mutant allele, because the disease is dominantly inherited. So the patients have a mutant allele and a wild type allele. And then the possible stoichiometries of a Kv1.1 containing channel are very complicated. You can have an abnormal Kv1 together with a normal Kv1, together with a Kv1.4 or Kv1.6, and so on. Lots of permutations.
And I guess, so I guess we might as well talk about drugs now, because you talked about channels. So sodium channels would be an obvious target for quieting down neurons, right? If you could block sodium channels. And that's some of the, so I guess talk about some of the commonly prescribed drugs.
Yeah, so I guess you can go quite a long way in epileptology just thinking of it as an excitation inhibition imbalance. Although we could talk about the limitations of that concept later on. But if we think of excitation as being sodium channels and glutamate receptors, and inhibition being potassium channels and GABA receptors, then actually quite a few of the anti-seizure drugs that are in use in the clinic target one or other of those of that family. So you have a lot of sodium channel targeting drugs, phenytoin, carbamazepine, lamotrigine, lacosamide. These are all drugs which facilitate inactivation or prevent recovery from inactivation. They slow down the recovery from inactivation. So they tend to diminish the function of the sodium channel. And then you have a lot of drugs that work on GABA receptors, and obviously the barbiturates and the benzodiazepines target GABA receptors, and they've been around for a long time. And in fact, we still use barbiturates and benzodiazepines in the clinic. Obviously, they have problems because of tolerance and rebound effects when you try to wean patients, especially the barbiturates. And then the glutamate receptors. So perampanel is a drug that was introduced relatively recently, which acts on AMPA receptors. So it inhibits AMPA receptors. Going back to the GABA receptors, of course, those drugs will facilitate the function of GABA receptors. So benzodiazepines and barbiturates will facilitate GABA receptor function. And then finally, potassium channels. So there was a drug, retigabine, which was actually terminated. It was pulled because of side effects, unpredictable side effects on the bladder and also skin discoloration. But that acted on a potassium channel, and it facilitated the opening of a potassium channel. So you have these inhibitory and excitatory mechanisms, and a lot of drugs will target one or other of those. The exceptions are a few drugs where we still don't know exactly how they work, such as valproic acid or sodium valproate, in use for many decades, but we still don't really understand. There's a lot of theories about how it works. Levetiracetam or Keppra is a very useful drug, very good drug, because it's relatively free of side effects and doesn't have lots of drug-drug interactions and so on. And that seems to work most likely by binding to a presynaptic protein called SV2A, but exactly how it works is also a little bit unclear. And there are a few other drugs where, again, we don't really understand that much about their mode of action.
And so you mentioned the one drug that I guess activates potassium channels. We played around with a drug called diazoxide. That's a potassium channel opener, and found it was neuroprotective in some of our models that involve excitotoxicity. But one issue with any of these drugs that affect excitability is excitability is critical for learning and memory and so on. So when you start messing around with these, I guess you got to get the dose just right so that in the case of epilepsy, you can suppress the seizures without having any major impact on cognition or other aspects of brain function.
Oh, absolutely. And the limitation of anti-seizure medication is mainly because the drugs work on the entire brain, in fact, the entire body. In fact, diazoxide, I think, doesn't it cause diabetes? Doesn't it affect insulin release?
At higher doses,
yeah.
we use
so effectively bathing the brain in a drug and hoping that it's going to affect the excitation inhibition imbalance in the epileptogenic circuits, but affect the rest of the but not affect the rest of the brain. The reality is that apart from some very, very rare examples, there is no such thing as a unique target in terms of a molecular target that is specific to epileptogenic zones or the neurons that contribute to seizures. Generally we think of epilepsy as a circuit imbalance, a circuit dysfunction, and so you're inevitably going to have a very narrow therapeutic window. So that's the main limitation, and as a result, about a third of people with epilepsy continue to have seizures despite optimal medication. So pharmacoresistance is extremely common. And in fact, that figure of about a third of people hasn't changed in decades. There was a recent study that showed that if you looked at people over a sort of 30-year difference, so going back 30 years when this was analyzed, and then looking at a recent cohort, the rate of pharmacoresistance hasn't changed despite the introduction of lots of new drugs. So the new drugs are, in a way, easier to use, fewer side effects, but ultimately you're still dealing with a very, very common problem, which is people who continue to have seizures despite medication. And that's where some of my recent work, together with colleagues trying to develop gene therapy for epilepsy, comes in, because we thought, well, in order to address this enormous unmet need, we have to come up with something different.
And so as far as other approaches, though, for example, ketogenic diet or fasting, we did some work with intermittent fasting, which increases ketones in the blood, and that suppress seizures. And we haven't talked about seizure-inducing toxins that are actually, there's a lot of interesting stories there where there's incidences where these people, for example, up in Canada, some people ate at the same restaurant and several of them developed seizures right after that. And it turns out they'd all eaten shellfish, and there was kind of it was a dry year, kind of a red tide year. So these algae that the shellfish feed on they actually produce the domoic acid. And as you go up the food chain, right, things concentrate. So they concentrated. Then these people ate the shellfish, developed seizures, and several of them actually had amnesia permanently. I think one of them passed away, and they actually looked at his brain, and there's a lot of damage to the hippocampus. And you and I and others, use these naturally occurring excitotoxins to induce seizures. So, and these are some of the models. Maybe you want to talk about some. Those aren't necessarily the best models.
No, but you're right to identify a very important group of models, which domoic acid, as I understand it, acts in a similar way to canic acid. It's another one of these algal toxins, and we use that model quite a lot. It's a very helpful, it's a very informative model because it can be used to induce a very good model of temporal lobe epilepsy. So temporal lobe epilepsy is, well, again, as the name indicates, all it means is that seizures arise from the temporal lobe, and patients can have a combination of different types of seizures, typically with disturbed consciousness, but often with automatisms. They'll act out some scenes. They'll have certain postures. Sometimes they only have clouded consciousness. They often have olfactory hallucinations. Sometimes they pluck at their clothes. So a whole lot of sort of interesting seizure manifestations in temporal lobe epilepsy. But a common structural correlate is hippocampal sclerosis. So people have loss of CA1 pyramidal neurons, loss of CA3 pyramidal neurons. They have dispersion of the granule cells in the dentate gyrus, and they often have sprouting of mossy fibers and loss of certain subsets of interneurons in the hippocampal formation. So the way that we and a lot of other labs working on epilepsy models induce a model of temporal lobe epilepsy is to inject canic acid into the brain. You can also do it systemically, but if you inject it into the amygdala or into the hippocampus, you can induce very, very severe seizures. So like a status epilepticus, where the rodent goes into uncontrolled seizure for a period, and then you can terminate the seizure using a benzodiazepine, for instance, and then the animal recovers, and then it starts having spontaneous seizures, which can be confined to the hippocampal formation or can generalize. And if you then sacrifice these animals and look at the hippocampus, you get very, very similar patterns of histological damage as you see in the patients, with loss of CA1 and CA3 pyramidal neurons, granule cell dispersion, mossy fiber sprouting, loss of some subtypes of interneurons. So it's actually a very helpful model because it has that sort of face validity. Now, of course, if you ask the question, well, does it have construct validity? Well, domoic acid poisoning or canic acid poisoning is a pretty pretty infrequent event, but actually you can induce status epilepticus using other methods experimentally, and in humans, status epilepticus can arise from viral infections such as herpes simplex, encephalitis, or various other insults, and that can itself be followed by similar patterns. So there's a point of convergence there. Lots of different types of insults can lead to hippocampal damage, manifesting with temporal lobe epilepsy. So that's one model that we've used quite extensively. But we have recently looked at other models of epilepsy, and we've been very interested in something that's also been sort of inspired by genetic observations in humans, but quite different. These are somatic mutations. So it turns out there's a condition called focal cortical dysplasia, where patients have an area of the brain where there's a malformation of cortical development associated with malformed neurons, typically giant cells, and loss of the gray-white matter differentiation. They have abnormally migrating neurons. And this can be detected on an MRI scan. And it often presents in childhood with seizures. And if it's extensive, it can also be associated with some autistic features and intellectual disability. And the seizures can be very pharmacoresistant. So it's a very important problem. And in fact, one of the few treatments you can offer to people with pharmacoresistant epilepsy is surgery. And when children undergo epilepsy surgery, the commonest abnormality that's found on histology is focal cortical dysplasia. So that's the commonest in children, and the second commonest in adults after the hippocampal sclerosis type of temporal lobe epilepsy. So it turns out that FCD is typically caused by somatic mutations. So these are mutations that are not inherited. They're happening during the development of the brain, and you get a clonal expansion of cells carrying the mutation. And these mutations, it turns out, tend to affect genes that are converging on the mTOR cascade. So the mammalian target of rapamycin or mitochondrial target of rapamycin. So there's a point of convergence there. So there's genes, about 15 different genes have been identified as carrying mutations in FCD that all converge onto the mTOR cascade. So we've been modeling this in the lab. So a number of other laboratories have developed mouse models of FCD using in utero electroporation. So you inject a plasmid carrying a gene in the ventricle of the embryo, and then using an electric force across the uterine horn, you push this plasmid into the developing brain, and then after the mouse is born, you then screen it for the fluorescence reporter that you can co-inject with the plasmid, and then these animals will start having seizures. So we've implemented this model recently, and it's a very useful one because it has face validity. These mice have seizures. They have behavioral abnormalities that recapitulate some of the abnormalities in the children, and it has construct validity because we're using plasmids which will also overactivate the mTOR cascade. So that's an interesting alternative model.
So the mTOR cascade, well, generally in protein synthesis. So then when it's activated, the just levels of general overall protein synthesis go up. So the idea, I guess, then is that if mTOR is abnormally active during brain development, it may be accelerating development. Development normally proceeds in kind of a controlled way, but maybe it's happening too fast. And this is one thought, I think, in autism too, right? That there's, like, as you mentioned, that accelerated growth that leads somehow to, as synapses form, an imbalance in activity.
Yeah. I mean, it's a very interesting field. I mean, of course, tuberous sclerosis is another mTORopathy, and the dominant theory there is that it's a two-hit phenomenon. So you inherit a mutation in the TSC1 or TSC2 gene, which then predisposes to a lot of the manifestations of tuberous sclerosis, but then a second hit can then give rise to the tubers and the giant cell astrocytomas and the other manifestations. And hemimegalencephaly is another malformation of cortical development, sometimes associated with very pharmacoresistant epilepsy, as well as intellectual disability and autistic features. And there you have an entire hemisphere that's affected. So that may be part of the same continuum of mTORopathy. So at one end, you have these very, very focal cortical dysplasias, and at the other end, you have an entire hemisphere affected. But of course, in autism, if you look at it from a different perspective, I mean, here we're talking about these malformations of cortical development with a very obvious structural abnormality on the MRI scan. But if you look at autism in general, then there's been an enormous literature over many decades of careful neuroimaging, finding subtle abnormalities. Again, I think of autism as a point of convergence, and lots of different things that can go wrong. But so actually putting one's finger on it is difficult, and so we know that genetics is important. But to what extent somatic mutations that are not associated with an obvious structural abnormality contribute to autism is the big unknown. I think that's a very, very interesting area that we're going to see a lot of exciting breakthroughs in the next few years of trying to understand to what extent somatic mosaicism explains some major psychiatric disorders.
So obvious question, have you tried rapamycin, an inhibitor of mTOR, in your model?
We haven't, but other people have. And we know that these rapalogs, these rapamycin analogs, are partially effective in treating the epilepsy associated with some of the mTORopathies. There have been some trials in children with tuberous sclerosis, and they are partially effective. But again, there are limitations to the use of these rapamycin analogs because they affect the immune system, and they can't be used at very, very early stages of development.
So the rapalogs, I guess I don't know this for sure, but my assumption is rapamycin is no longer patentable. So you modify it a little bit, and it has the same action as rapamycin. You call it a rapalog, and then if you get approval, you can charge a lot more higher price and say it's better than rapamycin.
I don't know. There are these drugs, everolimus and sirolimus. I don't know. I don't know.
Yeah. Okay. I don't know. I don't know. I've kind of seen this over the years, right? And certain drug in neuroscience, certain drug comes out, say a serotonin reuptake inhibitor, and then that one goes off patent. So company makes another one, and then they don't compare it head to head to the previous one in the clinical trial. They just test it versus placebo. And anyway, this is one of my pet peeves in pharmacy. Let's get back to, so diagnosis of epilepsy in general, the electroencephalogram is like a critical thing, right? If you can pick up, and a lot of times they'll be, when a person is not having a seizure per se, there's still some abnormalities. You want to discuss that a little bit?
Yeah, so well, I have to admit, I am not a clinical neurophysiologist. I'm not an expert in reading EEGs, but of
Uh,
course, it is a very, very important.
well, what do you think is happening at the local circuit level?
Okay, all right. So, well, okay, I'm not going to try to speculate about exactly how many dipoles need to align and so on. But no, I mean, you are basically looking at the population activity with a relatively low spatial resolution. So temporal resolution is not bad, although the skull and skin act as a low pass filter. So you don't really get that much insight into what's happening above about 50 hertz or thereabouts. And also typically the EEG is high pass filtered. So you don't really know very much about what's happening at very low frequencies. So there's a lot of theoretical and experimental literature that's just starting to be put together on, the hidden, the iceberg under the water, which is the DC shifts, the very, very low frequency shifts that probably underlie a lot of the interesting phenomena that are associated with seizures. That the cortical spreading depression is obviously something that we think of in terms of migraine aura, but cortical spreading depolarization or depression has a complicated relationship with seizures. For instance, after a seizure, there is sometimes a period of electrical silence, the post-ictal depression. To what extent that actually represents a spreading depolarization in an area of cortex is an interesting question, which I don't think has been resolved. But anyway, so coming back to the diagnostic role of EEG, in certain syndromes, it can be very important, especially in some childhood syndromes. There's some characteristic patterns. In adults, it's very useful to capture a seizure, but of course, seizures don't happen very often. And so there are some provoking stimuli that sometimes are used. So sleep deprivation or hyperventilation or looking at flashing lights. There's a certain photoparoxysmal response which can be very useful in detecting either a seizure or detecting a vulnerability to seizure. So, for instance, a photoparoxysmal response is not generally thought of as a seizure, but it is an abnormality which is seen in people and is associated with a predisposition to seizures, especially in the occipital lobe. But an interesting phenomenon is also, and also I should point out that people with primary generalized epilepsy sometimes have a characteristic spike wave discharge. So the classical childhood syndrome of childhood absence epilepsy is quite an amazing phenomenon because these children, typically children, they usually grow out of it, but sometimes they carry on, that they will suddenly flip into a generalized seizure, which is not typically associated with motor convulsion. And it's an absence. They have a sort of, they just sort of zone out for 10, 20 seconds or longer. And during that time, they have this characteristic spike wave discharge, typically about 3 hertz. And then they just return to normality after that. And it raises interesting questions about what's actually the nature of working memory, for instance. There are these extraordinary videos of children reciting the alphabet and suddenly they stop and they just carry on where they left off. Anyway, that's a digression. So in terms of focal epilepsy, which I guess is closer to what I've been studying more recently, if you don't capture a seizure, you can still sometimes capture what are called interictal discharges, which are a biomarker which is associated with focal seizures. And these are very brief discharges, sort of spike or sharp waves on the EEG. And the nature of these interictal discharges has attracted a lot of interest recently. And it's something that we've been very interested in because if you induce a focal model in a rodent, in an awake rodent, you can see certain interesting patterns. And to what extent these patterns that we're seeing in these mouse models actually recapitulate what happens in the humans, of course, can be discussed. But what we found is that if we look at calcium signaling in neurons, or GABA signaling, or glutamate release, using fluorescent reporters, we see some very interesting patterns. So we can do this by inducing seizure-like phenomena by just putting in a chemoconvulsant in an area of the cortex. But we're trying to do this also with spontaneous seizures. And we can see a number of patterns. So for instance, with a seizure a proper seizure that spreads across the cortex, we see a pattern of evolution of activity that spreads from the focus where we put in the chemoconvulsant, and it spreads across the cortex. And there are other routes of spread which are subcortical. And so lots of interesting patterns there. But when we look at in between seizures, we sometimes see little discharges which look a bit like aborted seizures. And we think that's mainly the brain's last-ditch attempt to suppress the escape of the excitability across the cortex. A little bit like a fire in the kitchen, where you have a fire blanket that you put onto the chip pan. So when you have a fire, you reach for the fire blanket and smother the fire, and that's a bit like a wave of glutamate that gets released. But intermittently that fails, and then the seizure escapes, and then your whole kitchen goes up in flames. So that's the sort of analogy I like to use. And so we've been looking very closely at what actually happens with GABA and glutamate using these fluorescent reporters for these neurotransmitters. So iGABA sniffer and iGlu sniffer. They were developed by Jonathan Marvin and Lauren Luger at Geneva Farm. And these have been very powerful tools to look at what's actually happening at synapses. And what we see is that indeed there's a wave of GABA as well as a wave of glutamate with these intraictal discharges. But the GABA seems to be in a ring around the focus, smothering it. And every so often, it's just not sufficient to smother it. And then the excitability escapes and spreads across the cortex. Why that happens is not entirely clear, but this idea of the breakdown of inhibitory restraint has been around for a few decades. And there are a number of mechanisms that people have speculated about. So for instance, it could be that the GABAergic interneurons that release the GABA fail to release the GABA, perhaps because they run out of GABA vesicles, or perhaps because the interneurons themselves become over-depolarized and they can't, the membrane potential can't return to rest, to fire again. They just stay depolarized and they eventually stop firing. Another possibility is the GABA is overactivating the GABA-A receptors, and then so much chloride goes into the neurons through the GABA-A receptors that it collapses the driving force for chloride, and then the GABA receptors fail to inhibit the postsynaptic neurons. There's another hypothesis, which is that you load the GABA receptors open so much that chloride goes into neurons, and then eventually chloride has to be pumped out of neurons, and the transporter that pumps chloride out of neurons, KCC2, the potassium chloride cotransporter type 2, it brings potassium out of neurons together with the chloride, and then the extracellular potassium concentration accumulates, increases, and eventually that then has a depolarizing effect on other neurons. So there's all these theories about how inhibitory restraint fails, and some of our observations have provided some support for one or other of these models. I suspect that all of them are correct in different situations. I don't think any of these models has to be shot down. It's just a question of which one is most important in which type of epilepsy or at which stage of the whole process. But anyway, seizures are quite fascinating. And it is quite remarkable the brain can crash in all these different ways. You know, it can crash into a seizure, it can crash into spreading depression, and then you get a migraine aura, or it can crash into a paroxysmal movement disorder. So that it's an endlessly fascinating topic. You better stop me there, otherwise I'll just carry on.
We had before I retired from the NIH a few years ago, in the preceding few years, I had a couple of postdocs that had some really interesting findings relevant, I think, to this discussion. There's some mouse models of Alzheimer's disease where there's a lot of this amyloid accumulating in the brain. They just spontaneously develop seizures and even die from them. So we had some of these mice and we recorded EEG, and we were manipulating. I mentioned the intermittent fasting, which actually the first model that we found that this, if intermittent fasting was neuroprotective was in the mechanic acid seizure model. But anyway, so these mice get a lot of amyloid, they develop seizures, and we see a lot of bursting activity. And then we videotaped the cages too, so because some of them were dying, and so we could have the EEG recording just before they died. And it went really crazy then. But so we also supplemented the diet with beta hydroxybutyrate, ketone, and then that suppressed the seizures. And then I had a postdoc who's really good patch clamper, and he recorded what he did is maintain rats on intermittent fasting or give ketone, and then prepare acute slices, right? And record inhibitory postsynaptic currents from CA1 neurons. So these are essentially GABA being released onto the receptor, which is a chloride channel. And what he found was that the intermittent fasting and the ketone enhance GABAergic tone. And then the story gets more interesting. We were doing a lot of work with a certain protein in mitochondria, an enzyme called SIRT3, which is a protein deacetylase that removes acetyl groups from lysine residues. Anyway, it can affect the function of proteins in a manner analogous to phosphorylation. And so there's a lot of evidence that SIRT3 is really important in the life of mitochondria and in energy metabolism, but also in other things. So a colleague at NIH had made SIRT3 knockout mice, and so we found that when we crossed the SIRT3 knockout mice with the Alzheimer's mice, those mice developed seizures really early and died. So to exacerbate the seizure phenotype, and so kind of the bottom line is there's some link between the mitochondria and the synaptic regulation of synaptic excitability. And then last thing along this is that the GABAergic neurons, they're just packed with mitochondria if you look at them. And the reason is they fire a lot, right? They're really active, so they need a lot of mitochondria to have the ATP so they can restore the ion gradient. But I guess having said all this, there might be some interesting work in the future, like even from a preclinical standpoint of targeting mitochondrial function. Of course, ketones target that too, right? Because they're alternative energy source.
Yeah, absolutely. And there are certain mitochondrial cytopathies that cause seizures, or seizures are a manifestation of these disorders. Yeah. And of course, when we talk about interneurons, they are extremely heterogeneous. So of course, the parvalbumin positive fast spiking basket cells will, as you said, I like to think they're built for speed. But there are other interneurons that have other functions and other properties. So I'm always a little bit nervous about generalizing too much about interneurons. So when I talk about this sort of inhibitory surround, I'd like to really drill right down to find out which interneurons are doing this. But if you get to the ketogenic diet is very interesting.
The bottom line, if there weren't any GABAergic inhibitory neurons, we wouldn't have any brain. We'd be a continuous seizure.
Yes, and of course, status epilepticus is something that I see every so often as a clinician, because one of the things I do is some ward rounds on the neurocritical care unit, and we do sometimes see patients with status epilepticus, and sometimes we never find the cause. And although many patients with status epilepticus will come out of status and sometimes are left with epilepsy, we have lost a few patients over the decades, and exactly what the reason why they develop status epilepticus out of the blue is quite mysterious. So there is something called NORS, new onset refractory status epilepticus, and whether it's antibody driven or some sort of T cell immunity or some sort of metabolic disorder is quite unclear. Some of these patients have been completely normal until they suddenly develop status epilepticus in their 20s or 30s. And in the old days, I remember, I mean, I've been around so long that I remember when we had mysterious neurology cases, we'd say, well, it must be mitochondrial. Nowadays, we tend to say, well, it must be an antibody we haven't found. But yeah, there's still a lot of that. Well, fortunately, these are very, very infrequent cases, but I have seen a few. But there's still a lot to be discovered. Yeah.
Okay, so in the last part of this, let's talk about two recent papers. One that I learned about before we started this podcast, but the other one, which I looked at a lot, was Andreas Lieb is the first author, Nature Medicine paper in 2018 on, you mentioned early on, you mentioned throughout this that, treating with, or we talked about how you treat with drugs, you're bathing the whole brain in the drug, or if you do gene therapy that affects the whole brain, you may get adverse effects. So in the Nature Medicine paper, your idea was, okay, you're going to have a gene therapy based mechanism where the gene essentially only turned on when the seizure is starting, I guess. Is that?
Well, the paper that we published in Nature Medicine, so Andreas Lieb's paper, so that's based on using a glutamate gated chloride channel. So previously we'd shown that we could suppress seizures using constitutive overexpression of Kv1.1, our favorite potassium channel, and we could also use a chemogenetic approach using muscarinic DREAD. So these are these mutated muscarinic receptors that design a receptor exclusively activated by designer drugs. That's a DREAD acronym developed by Brian Roth and his colleagues. So we used a DREAD. This was work by Dennis Ketzel earlier on, where we express the DREAD in the seizure focus, and then nothing happens until you give the drug that activates the receptor, right? And we showed an on-demand anti-seizure gene therapy. But of course, that requires an exogenous ligand, the drug to activate the receptor. And since then, we've shown that we can use olanzapine rather than clozapine N oxide. But anyway, that's something that's potentially amenable to clinical translation. But when Andreas was in the lab, when Andy was in the lab, he came up with this idea that we could use an ion channel that responded to the excitatory neurotransmitter, but had an inhibitory effect, right? So typically glutamate acts as an excitatory neurotransmitter, and we know that glutamate goes up in seizure foci, and in fact, with seizures, glutamate goes very high because lots of excitatory neurons are firing together, and the glutamate uptake mechanism is overwhelmed. So there's evidence that glutamate goes up during seizures in seizure foci. So we thought, well, if we could find a receptor that was activated by glutamate but had an inhibitory function, then that could act as a gene therapy tool. And so there is such a receptor. In worms, in C. elegans, there's a receptor called GluCl, which is a glutamate-gated chloride channel. And in fact, it was sort of indirectly in the news during the COVID pandemic because certain famous people were talking about ivermectin being a useful COVID drug. It turns out ivermectin is an agonist of GluCl. Now, in fact, at the beginning, we thought, well, why don't we just use ivermectin with GluCl as a conventional chemogenetic pair? We express GluCl, and then we give ivermectin, and it would act as an anti-seizure conventional chemogenetic approach. There are a couple of problems. One is that ivermectin actually gets across the blood-brain barrier very, very slowly, and ivermectin itself acts on GABA receptors, which complicates things even further. But anyway, so Andy had this idea that if we actually tweak the GluCl itself to make it more sensitive to glutamate, because the wild type GluCl in C. elegans has quite a high EC50 for glutamate. It has to have hundreds of micromolar glutamate in order to open. So what we did was we dialed down the EC50. So we made the gluCl much more sensitive to glutamate, made the EC50 come down to about 10 micrograms.
Was that done in just like a trial and error? You mutated?
Well, no, there was actually some data from Caltech so there was Henry Lester's laboratory that they had actually done some work and they had shown that there are some amino acids that if you manipulate these, you can alter the glutamate sensitivity, you can alter the ivermectin sensitivity. So we investigated this, reproduced some of their work, and then showed that if we use a particular combination to lose the ivermectin sensitivity, but increase the glutamate sensitivity, then we could achieve this tool, which would act as an endogenous sort of closed loop system. So if these receptors were expressed in the neurons, they would detect glutamate and then cause the chloride channel to open and then shunt the excitatory current and then have an anti-epileptic effect. So this worked. And we showed it to work in an epilepsy model, which we haven't talked about so far, which is using tetanus toxin, which is a toxin which cleaves one of the snare proteins. And that has an effect on inhibitory neurotransmitter release. But after the toxin's been cleared, these animals continue to have seizures for several weeks. And so we used this model to test to do like a preclinical trial. And that worked. There are some problems. We're using two quite bulky proteins, so the genes can't fit into an adeno-associated virus, which is the standard viral vector that most people use. We had to put it into a lentiviral vector, which is derived from HIV. I mean, lentivirus is still used quite extensively in ex vivo gene therapy, and there has been one trial of an in vivo clinical trial of a retrovirus related to lentivirus, but is not an ideal tool for doing gene therapy in patients. So we're still working on this, and we have some more work that we will be shortly wrapping up for submission for publication, where we're trying to shift this into an AAV system. But anyway, that was one approach, using the sort of closed loop mechanism whereby the system would detect a seizure and then inhibit the circuit in closed loop. The other approach that we published very much more recently was, I have to give credit to my colleague Gabriele Lignani, who had this idea that, so here's the challenge. If you do gene therapy, you can identify the seizure focus, in the clinic, because we're not in the clinic yet, but we hope to be in the clinic quite soon. You can identify the seizure focus using scalp EEG, intracranial EEG, using electrodes that you put into the brain or subdural grids, and obviously complemented by imaging and knowing what sort of seizures patients experience. So you can sort of work out where the seizures are coming from reasonably accurately, down to a few cubic centimeters. And then you can do the gene therapy in that area. And using the natural properties of the viral vectors that carry the gene, and also the promoters that you put into the construct that goes into the viral vector, you can bias expression towards one cell type or another. All right, so there are some promoters which are biasing expression towards excitatory neurons. We use the CAM kinase promoter quite extensively. So there's a calcium calmodulin kinase 2 alpha, which biases expression towards excitatory neurons. It's not perfect. It does leak into inhibitory neurons as well, but it doesn't seem to go into glial cells. But of course, when you're doing that treatment, you're still treating neurons, whether they're participating in seizures or not. So what Gabriele suggested was, well, what if we used activity-dependent promoters, which will sense the activity of neurons to drive the transgene expression? So there are some activity-dependent promoters. These are promoters of immediate early genes, such as CFOS. That's one of the famous ones. So CFOS lights up, it switches on massively in response to a seizure. And so we took the CFOS promoter and put that in the viral vector and drove our favorite potassium channel, Kv1.1. So if the neurons are overactive, the CFOS promoter gets switched on. That then drives the expression of the potassium channel, but only in the neurons that are participating in the seizure. So that should leave unaffected bystander neurons, which may have taken up the viral vector, but will not, if they're not participating in the seizure, the CFOS promoter won't be switched on, so the potassium channel won't be expressed. So it's only the neurons that are overactive that get dialed down because the potassium channel is overexpressed, and that causes a damping down of their excitability. So anyway, it worked, and it was well tolerated. We showed that if we did loads of behavioral studies in these mice where we treated the hippocampus, they performed perfectly normally in all the trials, all the tests that we threw at them. And we even put this into human cerebral organoids, these mini brains in a dish. We managed to reconstitute a sort of assembloid of inhibitory and excitatory neurons sort of growing together. And we elicited seizure-like phenomena using a particular cocktail of drugs, and then showed that the organoids which were treated with this activity-dependent promoter driving the potassium channel were relatively protected to a second insult.
Yeah, this is fantastic. So that's your science paper from last year, right?
Yeah, so that came out in Science in November, and the first author is Yichen Chu, Q-I-U.
Yeah, and yeah, you didn't mention surgeries, but I think it's less common, but still not, patients who are refractory to drugs or other treatments, ketogenic diet, whatever, surgery is still done to remove this epileptic focus. And so that's obviously quite invasive, but this method you're talking about is much less invasive, that I would think, because essentially you just need an injection needle to get the virus where.
Yeah, so I mean, epilepsy surgery is offered to a small proportion of people with focal refractory epilepsy. So people who continue to have seizures despite medication, in whom you can identify the seizure focus. And the typical pathologies are malformations of cortical development and hippocampal sclerosis. Those are the typical sorts of pathologies, but there are lots of other things, slow growing tumors, other types of malformations. Obviously, people with vascular abnormalities and tumors, there's an indication for surgery in its own right. And sometimes people with without an obvious structural abnormality are sometimes offered surgery. Surgery, of course, is destructive, it's irreversible, and it leaves a scar. And we know that penetrating brain injuries are an important cause of epilepsy. And so it's a bit paradoxical. You take out something, but you leave a scar, but you know that the scar is something that can cause epilepsy. But surgery is effective. And people with mesial temporal sclerosis, so people with hippocampal sclerosis, they have a rate of seizure freedom of above 50%, somewhere between 50 and 70%. People with foci in the frontal lobe, typically lower success rate. And there is a delay in getting to surgery. So a lot of these patients will have spent many years trying different combinations of medications before they finally undergo surgery. And it is thought that if you delay the surgery in children too long, it may not be to their benefit. It may be that we should be operating earlier. It is, of course, something that is not undertaken lightly, and a lot of people who are offered surgery actually decline it. They say, I don't want to have the surgery once now that I realize that this is irreversible, there is a risk, small risk. So some people opt for less invasive procedures, such as something called laser interstitial thermal therapy. So it's a type of laser delivered down a probe to burn out an area of the brain where the seizures are coming from. It's less effective, but it's less invasive, and some patients undergo that, and then if it doesn't work, they can still have the surgery, the definitive sort of resection. Gene therapy is also invasive, but it's similarly invasive to laser interstitial thermal therapy. It involves 1 or 2 burr holes, so small holes about the size of a dime, and then insertion of a cannula into the brain, and then slow infusion of the gene therapy vector. We've done some estimates. We think we could probably cover 2 or 3, using 2 or 3 trajectories, we could cover most typical foci. Obviously, some patients have multiple foci or have very, very extensive epileptogenic zones, and there we haven't really, sort of done the modeling yet. But we hope to start a clinical trial in the next year or 2. I've been saying that for about 5 years. I've been saying it's a year away for 5 years. Now I'm saying it's 6 months away, but it's still, anyway, in reality, it's still a year away.
Well, 2 years were destroyed by COVID, right? I mean,
yeah. But what we would do is typically we would recruit people at the point for a clinical trial at the point where they've already been evaluated for surgery, for surgical resection. And if they've had the investigations, which involve MRI, EEG, sometimes intracranial EEG, they've been seen by the surgeons, the neurologists, the clinical neurophysiologists, the psychiatrists, the psychologists, and everyone says, yep, this patient will be suitable. We can offer them this sort of risk and prospect for seizure freedom. At that point, we would then say, well, would you like to participate in a clinical trial? Because typically, especially in the hospital where I work, between the time they've had their pre-surgical workup and they come in for the surgery, there's usually a gap of weeks or months. So during that period, we would say, well, would you like to participate in the clinical trial? We would do the gene therapy into the region of the brain that would be taken out, right?
Yeah.
And so we bring them back. They have this relatively small procedure, gene therapy. And then if it works, they don't need to come back for the surgery. If it doesn't work, we take out the tissue at surgery, and then we look at it. And so we should have like a win-win situation. We either get the tissue or we get evidence of efficacy. And if the gene therapy hasn't worked, is it to do with dosage? Is it because we missed the target? Is it because we induce some sort of unexpected inflammation or whatever? Yeah. So we hope that the tissue would be informative.
Yeah, that's really exciting study. I look forward to that. It'll be fantastic if it works. Okay, so I know you're busy, and I won't take any more of your time, Dimitri, but I appreciate your willingness to do this, and I hope the listeners and viewers find it interesting. I'll put in the description section links to maybe a couple of review articles, and then for sure the Nature Medicine and Science papers. And
well, thank you, Mark. It was a real pleasure.
Yeah. Take care. Actually, the previous podcast I did was with Tom Foltini, because actually these GLP-1 receptor agonists that he uses was developed where I was.
Right.
Well, we helped a little bit. My lab helped a little with that.
It's a very interesting field. Yeah.
Yeah.
I hope it transforms the treatment of Parkinson's.
Yeah.
We'll have to see. Okay. Thank you very much, Mark.
Yeah. Cheers.